Right, okay. What I want you to do today is I want you to go to Lionel and go to the second lesson of topic 4, which is human influences. And some of it's going to be revision in terms of DNA, and some of it's going to be looking at how we can look at DNA for the in how we can use it in the future. So on there, with the first thing that's on there, there is a PowerPoint. I want you to flick through that. It goes things about like what is DNA, um, what does it stand for? So you should know it's deoxyribonucleic acid. And then it's asks you what it does. So what is our DNA there for? Now, generally our DNA just codes for proteins. Um, it strings your amino acids together to make proteins in your cells. So it's going to be like making muscles. So obviously, like that. That's obviously a bit of an extreme in case of muscles, but obviously it makes keratin, which makes your nails grow. Also, same for your hair. Makes all the different structures in your body from the DNA coding for different types of amino acids, which makes proteins. And it does that because it's got the base pairs. Now, when we watched one of the videos with the mitosis sisters, they might not be the mitosis sisters, we're going to call them that for now. They said that if you remember, it's the apples in the tree and the cars in the garage, A and T and G and C. So when you've got the base pairs of DNA, they will always be together. So A will always be with T, G will always be with C. So if you see a G, it should be a C next to it. If you see a C, it should be a G next to it. And that's just the base pairing of your DNA. Now, what they do is this bit you don't really need to know, but it just gives you a little bit of background. Is that every three base pair codes for a certain amino acid, and then those amino acids get like paired together, or get put together, and that makes your protein they transfer around. And that is just basically what a gene is. So, what is the thing about gene transfer is we take something we like in one species and try and put it into another. So like selective breeding, we were breeding over many, many generations to get what we want. In terms of this, we're just taking a gene for a characteristic that we like and we want to put it into something else. So in terms of gene transfer, we take a section of gene and transfer it to another living chromosomes, and that can be done in lots of different ways. And the most common one is using bacteria, and we use little things called plasmids. So in bacteria, you have plasmids, and what we can do is we can insert a DNA, some DNA that we want to usefully make something of, into that plasmid. So then this bacterial cell will then start producing that thing that we want. So one of the things what we have to do is we have to get the gene in there. So it's kind of difficult to get that in there, but the bacteria do swap plasmids from one bacteria to another. So they do swap genes really, really easily. So we just need to get the gene in there, and we use different bacteria as vectors. And a vector is just something that allows things to be transferring things. So like a mosquito is a vector for malaria. In the case of this, our bacteria is going to be the um, the vector for carrying the gene. So we can also use viruses as well to get the gene from one living thing into another, and that's called, let's say, genetic engineering. So if you click on, when you get onto, click on this link, and you'll be able to do it, see some examples of that. The most common example that you will need to know about is just using genetic engineering to make insulin. So we know what the gene is to make insulin is, we can cut that out, put it into the plasmid, and then we get bacteria producing lots of insulin, which is really, really quick because it's very, very quick, and we get human insulin made. Used to be that we'd have to have a pig, and you'd have to get the insulin from the pig, slaughter the pig, and then that would be, obviously that's not great. So obviously you're killing pigs. But then once you've done that, you can then put it in a large vat, and you can make large quantities of the insulin. And there are, like I said, there's advantages to this. You can make larger quantities. It's identical to human insulin, and we don't have to kill the pigs. And it's also cheaper to produce. Um, Now we've done this not only bacteria to produce insulin, but we're starting to do it with plants, and you'll see GMOs labelled in Hong Kong as genetically modified plants. So we've taken genes from like fish that live in really cold environments, and we put them in tomatoes so they don't freeze at low temperatures, so they don't like get damaged. We can change it to like there's other examples of like golden rice, which is probably down here somewhere. Golden rice increasing the vitamin A content of that. So basically, what I want you to do today is just go through this PowerPoint, and then once you have finished that PowerPoint, I want you to start looking at this next thing. So using the PowerPoint that I just went through, I want you to go on to Google Classroom. On there, you've got this worksheet. So I would like you to work your way through this first bit. Just remember what I've just said about the different parts of DNA. People had difficulty with this yesterday, coming up with what would match the other strand. So, for example, it says a C there, so that means I'd have to have a G, and then it says a G, so I'd have to have a C, I'd have to have a C, and then I'd have an A, because it's matching the other side. So that's all you have to do. So just keep it very simple and just work your way through this. And at the end of the lesson, can you submit that? I would also like you to do, I'd like you to go back to Lionel. Okay, and on Lionel, what I'd like to do is click on genetic engineering and just do the genetic engineering quiz so I can see how well which bits you get, which bits you don't know. Okay, I'll be on Google, I'll be on Google Meet all lessons. If you've got any questions, just pop them in the chat or email me. Yeah, so good luck with that, and I'll speak to you in a moment. Cheers.